Precise RNA Gene-Editing Technology. Introduction. The advent of precise RNA gene-editing technologies, particularly through the utilization of CRISPR systems, has revolutionized genetic research and therapeutic applications. These technologies facilitate precise modifications to the genome, enabling targeted interventions for various genetic disorders including cardiovascular diseases (CVD). This report provides a comprehensive overview of the advancements, applications, and challenges of precise RNA gene-editing technologies. Historical context and evolution. The foundation for modern gene-editing technologies was laid in the 1950s with the discovery of the double helix structure of DNA. Over the decades, various methods emerged to manipulate genetic material, including 1. Restriction enzymes 1970s to 1980s, enzymes that cut DNA at specific sequences, allowing for the development of recombinant DNA technology. 2. Zinc finger nucleases ZFNs and transcription activator-like effector nucleases TALENs 1990s. These introduced specific double-strand breaks DSBs in DNA to facilitate targeted editing. 3. CRISPR/Cas9 system 2012, a groundbreaking tool that simplified gene editing by utilizing an engineered RNA guiding the Cas9 nuclease to specific DNA targets, creating DSBs. 4. Base editing 2016, a technique that allows for the conversion of one DNA base into another without making DSBs. 5. Prime editing 2019, a more versatile approach enabling targeted insertions, deletions, and substitutions with high precision. This evolution reflects a trend towards enhancing the specificity and efficiency of gene editing technologies, addressing challenges such as off-target effects and delivery mechanisms. Advancements in CRISPR technology. Recent advances in CRISPR technology have led to the development of various tools and techniques aimed at enhancing genome editing. Novel Cas proteins, the discovery of new Cas proteins, e.g. Cas12 and Cas13, has expanded the repertoire of gene editing tools, allowing for greater specificity and efficiency. Engineered variants, high-fidelity Cas9 variants such as SpCas9HF1 and ESpCas9 have been developed to reduce off-target effects while maintaining on-target activity. Transposon-associated systems, the integration of transposon-associated CRISPR systems Cas allows for the precise insertion of larger DNA fragments into genomes, enhancing the capabilities of gene editing. Applications in cardiovascular research. CRISPR-based gene editing technologies have found significant applications in cardiovascular research. The ability to create genetically modified animal models has advanced the understanding of CVD and its underlying genetic mechanisms. Key applications include 1. Disease modeling. CRISPR/Cas9 has been employed to generate animal models that mimic human cardiovascular conditions, enabling researchers to study disease mechanisms and test potential therapies. For instance, the creation of mice with mutations in genes associated with hypertrophic cardiomyopathy (HCM) and dilated cardiomyopathy (DCM) has provided insights into the pathophysiology of these conditions. 2. Therapeutic approaches. Gene editing technologies hold promise for treating inherited cardiovascular diseases by correcting mutations at their source. Recent studies have demonstrated the successful correction of mutations in genes such as MYBPC3, which is implicated in familial HCM. 3. Base and prime editing. These technologies have emerged as promising therapeutic strategies, allowing for precise modifications without the complications associated with traditional CRISPR/Cas9 editing. For example, base editing has been used to convert pathogenic mutations in the PCSK9 gene, which plays a critical role in cholesterol metabolism, leading to reduced LDL levels. Challenges and limitations. Despite the significant advancements in RNA gene editing technologies, several challenges remain. Delivery mechanisms. Efficient delivery of CRISPR components to target tissues is crucial for successful gene editing. Current methods, including viral vectors and lipid nanoparticles, face limitations regarding tissue specificity and potential immunogenicity. Off-target effects. Minimizing unintended genetic alterations is a critical concern, particularly for therapeutic applications. Continued efforts to develop high-fidelity Cas variants aim to address this issue. Ethical considerations. The potential for germline editing raises ethical questions regarding the long-term implications of genetic modifications. Future perspectives. The future of precise RNA gene editing technologies looks promising, with ongoing research focused on 1. Improving delivery systems, developing safer and more efficient delivery methods for CRISPR components to enhance therapeutic applications. 2. Expanding targeting range, engineering new Cas proteins and optimizing existing ones to broaden the range of targetable sequences. 3. Clinical applications. Continued progress in translating laboratory findings into clinical settings for the treatment of genetic disorders, particularly in cardiovascular diseases. Conclusion. The evolution of precise RNA gene editing technologies, particularly through CRISPR systems, has significant implications for genetic research and therapeutic applications. Ongoing advancements in efficiency, specificity, and delivery methods are paving the way for innovative treatments for genetic disorders and cardiovascular diseases. As these technologies advance, addressing the associated challenges will be essential for their successful implementation in clinical practice. Counterintuitive information in precise RNA gene editing technology. The development of precise RNA gene editing technologies, particularly those based on CRISPR systems, has brought dramatic changes in genetic research and therapeutic applications. However, several counterintuitive aspects arise from the advancements and applications of these technologies. 1. Efficiency versus specificity. While one would assume that increased editing efficiency comes with improved specificity, this is often not the case. For example, CRISPR/Cas9 systems are known for their efficiency in generating double-strand breaks DSBs in target DNA. Yet they frequently lead to off-target effects. This paradox suggests that optimizing for one parameter may inadvertently compromise another, thereby complicating the quest for ideal gene editing tools. 2. Base editing limitations. Base editing technologies, heralded for their precision in making targeted point mutations without DSBs, ironically have a limited scope. They are currently restricted to specific types of mutations, CG to TA and AT to GC, which might lead researchers to overlook the vast array of other genetic modifications necessary for a comprehensive therapeutic approach. This limitation can be surprising given the initial enthusiasm for the technology's potential. 3. Therapeutic potential versus safety concerns. The excitement surrounding CRISPR-mediated gene therapy as a solution for genetic disorders is often tempered by concerns about safety and potential unintended consequences. Counterintuitively, while gene editing offers the promise of curing genetic diseases, the actual clinical application faces hurdles due to risks of off-target mutations and genomic instability, which may introduce new health issues rather than resolve existing ones. 4. Post-mitotic cells in HDR. Traditional understanding has indicated that homology-directed repair HDR is primarily effective during the S/G2 phases of the cell cycle, which poses challenges for gene editing in post-mitotic cells. However, recent findings have shown that HDR can also occur in these cells under certain conditions, challenging the notion that they are inherently resistant to this type of gene editing. This counterintuitive finding opens new avenues for therapeutic applications in non-dividing tissues. 5. Vector delivery challenges. It is often assumed that viral vectors such as AAVs are the most effective means of delivering gene editing components due to their efficiency. However, the size limitations of these vectors can paradoxically hinder the delivery of larger Cas proteins in donor templates, which are essential for certain editing strategies. This irony highlights the need for innovative delivery systems that balance efficacy and size constraints. 6. In vivo versus ex vivo applications. While in vivo editing is perceived as a more direct approach to treating diseases, it presents unique challenges such as immune responses and off-target effects which can complicate outcomes. Conversely, ex vivo editing allows for more controlled environments but requires sufficient viability and functionality of cells outside the body. The assumption that one method is universally superior is thus misleading. 7. Modeling versus treatment. The use of precise gene editing technologies in modeling diseases is often met with enthusiasm due to their potential to mimic human conditions accurately. However, the same technologies might not be as effective in therapeutic contexts, where the ideal conditions for editing do not always translate into successful treatments. This discrepancy challenges the expectation that advances in modeling will seamlessly lead to therapeutic breakthroughs. 8. Ethical implications and accessibility. The rapid development of gene editing technologies raises ethical concerns that may limit their application in clinical settings. Surprisingly, while these technologies promise significant advancements in medicine, their potential misuse could provoke public backlash and regulatory hurdles, thereby slowing down the very progress they aim to promote. 9. CRISPR as a double-edged sword. CRISPR technology is often lauded for its revolutionary potential in genetics, yet it also poses significant biosecurity threats. The ease and accessibility of gene editing tools could enable misuse, such as the creation of harmful pathogens or genetically modified organisms with unintended consequences. This duality complicates the narrative surrounding the benefits of gene editing. 10. Integration of new technologies. The integration of new gene editing technologies, despite their promise, often requires extensive validation and optimization. For instance, while prime editing offers remarkable versatility, its practical implementation in clinical settings may lag due to the necessary refinements and modifications. This creates a gap between technological potential and real-world applicability. In conclusion, while precise RNA gene editing technologies offer transformative potential for genetic research and therapy, they also introduce complexities and contradictions that challenge straightforward interpretations of their efficacy and safety. These counterintuitive insights underscore the importance of ongoing research and reflection on the implications of such powerful tools in genetics. Significant overlooked content in the report on precise RNA gene editing technology. In reviewing the advancements in precise RNA gene editing technologies, particularly those utilizing CRISPR systems, there are several critical aspects that many readers may overlook. Despite their significance in shaping the future of genetic research and therapeutic applications. 1. Technological evolution and novel Cas variants. While the report highlights the progression from CRISPR/Cas9 to more refined systems like base editing and prime editing, the emergence of novel Cas variants and their unique properties are often understated. For instance, the report mentions the discovery of miniature Cas proteins, e.g. Cas12F, and their potential for broader applications in genome editing due to their smaller size, which enhances delivery methods using vectors like AAV adeno-associated virus. This aspect is crucial as it not only expands the toolbox available for genetic editing but also addresses the challenges associated with delivering larger Cas proteins into target cells. Significance: understanding the variety of Cas proteins and their adaptations could lead to more efficient and specific genome editing approaches, particularly in therapeutic contexts where delivery to specific tissues is critical. 2. Challenges in HDR efficiency and off-target effects. The challenges associated with homology-directed repair HDR efficiency and the off-target effects of CRISPR systems are mentioned but require deeper exploration. The report touches on the fact that HDR is often less efficient than non-homologous end-joining NHEJ, which can lead to unintended mutations. However, the complexities of cellular environments and the varying efficiencies of HDR in different cell types or during different phases of the cell cycle are often overlooked. Significance: acknowledging these challenges is essential for researchers aiming to optimize genome editing strategies. Innovations aimed at enhancing HDR efficiency, such as the use of small molecule inhibitors that suppress NHEJ, are critical areas of ongoing research that could greatly improve the precision of gene editing. 3. Emerging applications in cardiovascular research. The specific applications of CRISPR technology in cardiovascular research as discussed in the report are significant but often overshadowed by broader discussions of gene editing technologies. The report outlines how CRISPR has been used to generate animal models for studying genetic mutations associated with cardiovascular diseases and to explore potential gene therapies. Yet the implications of these innovations on understanding and treating prevalent diseases like hypertension, cardiomyopathy, and atherosclerosis are profound. Significance. Diving deeper into these applications reveals the potential CRISPR technologies have in not only elucidating the genetic basis of cardiovascular diseases but also in developing targeted therapies that could dramatically improve patient outcomes. 4. Ethical considerations and regulatory frameworks. While the report concludes with a discussion on the ethical implications and potential misuse of gene editing technologies, it is crucial to expand on the necessity for robust regulatory frameworks. The rapid advancement of CRISPR technologies raises significant ethical dilemmas, particularly concerning germline editing and the implications for future generations. The discussion on biosecurity risks associated with gene editing, including the potential for creating virulent pathogens or bioweapons, is pertinent yet often inadequately addressed. Significance. Emphasizing the need for comprehensive guidelines and international cooperation in the regulation of gene editing technologies is essential for ensuring responsible research and application. As the technology evolves, so too must the frameworks governing its use to prevent abuse and unintended consequences. 5. Innovative delivery mechanisms. The report discusses various delivery mechanisms for CRISPR components but may not fully capture the breadth of innovative strategies being developed. For instance, the use of virus-like particles VLPs and lipid nanoparticles LNPs for delivering CRISPR components showcases a significant shift towards minimizing immune responses and enhancing delivery efficiency. Moreover, the advancements in dual AAV systems that allow for the delivery of larger constructs are pivotal for the future of gene therapy. Significance. Highlighting these cutting-edge delivery systems provides a clearer picture of how the challenges of delivering gene editing tools can be addressed, leading to more effective therapeutic interventions. 6. Potential of prime editing beyond genome editing. Prime editing has been heralded as a revolutionary advancement in the precision of genetic modifications. Yet its potential applications extend beyond traditional genome editing. The report mentions its ability to create various genetic modifications without double-strand breaks but does not delve into its implications for epigenetic modifications or other novel applications in synthetic biology. Significance. Understanding the broader applications of prime editing could inspire innovative approaches in both therapeutic and research settings, particularly in developing tools for controlling gene expression and creating novel genetic constructs. Conclusion. The report on Precise RNA Gene-Editing technology encapsulates a comprehensive overview of advancements and applications. However, by emphasizing the significance of often overlooked content, such as novel Cas variants, HDR challenges, cardiovascular applications, ethical considerations, innovative delivery methods, and the potential of prime editing, readers can gain a more nuanced understanding of the landscape of gene editing. This knowledge is essential not only for advancing research but also for responsibly navigating the ethical and regulatory challenges that accompany these powerful technologies. 1. Technological evolution and mechanisms. The advancement of precise RNA gene editing technologies, particularly CRISPR systems, has revolutionized genetic research. Key innovations include the evolution from traditional methods like ZFNs and TALENs to CRISPR/Cas9 base editing and prime editing. These new systems allow for targeted modifications with high precision and efficiency. A comprehensive understanding of the mechanisms underlying these technologies, such as homology-directed repair HDR and non-homologous end joining NHEJ, is essential for improving their applications in therapeutic contexts. 2. Applications in cardiovascular disease (CVD). The potential of CRISPR-based gene editing technologies in cardiovascular research is significant. These tools enable the creation of genetically modified in vitro and animal models to study cardiovascular diseases and facilitate the development of targeted treatments. The ability to model diseases like hypertrophic cardiomyopathy and atherosclerosis through precise gene editing opens new avenues for understanding disease mechanisms and testing therapeutic strategies. 3. Challenges in delivery and off-target effects. Although CRISPR technologies hold great promise for gene therapy, challenges remain, particularly in delivering genome editing components to target tissues effectively. The risk of off-target effects. Where unintended genetic modifications occur, poses significant concerns for the safety of these therapies. Addressing these challenges through innovative delivery methods such as using lipid nanoparticles or engineered virus-like particles is critical for the successful translation of these technologies into clinical applications. For future prospects and ethical considerations, the continuous refinement of genome editing tools aims to enhance their efficiency and specificity while minimizing off-target effects. As CRISPR technology progresses, ethical considerations surrounding its application, especially in human germline editing, must be addressed. The potential for misuse in creating harmful genetic modifications or bioweapons raises significant ethical and regulatory challenges that need careful oversight to ensure responsible use of these powerful technologies. These points highlight the transformative impact of precise RNA gene editing technologies on genetic research and therapy, while underscoring the need for ongoing innovation and ethical considerations in their application. Here are 3 famous historical cases that parallel advancements in precise RNA gene editing technology, particularly focusing on CRISPR and its implications in genetic research and therapy. 1. The Human Genome Project (HGP). The Human Genome Project, which began in 1990 and was completed in 2003, was a monumental international research initiative aimed at mapping and understanding all the genes of the human species. This project not only identified the sequences of the 3 billion base pairs that make up human DNA, but also paved the way for an immense leap in genetic research, including gene therapy. The HGP's success has been instrumental in advancing our understanding of genetic disorders and has set the groundwork for precise gene editing technologies like CRISPR. With the ability to edit genes at a specific location in the genome, CRISPR allows for targeted therapies that can potentially correct genetic defects, similar to the HGP's goal of understanding the genetic basis of diseases. 2. Gene therapy trials, e.g. SCIDX1. The first successful gene therapy trial for a genetic disorder occurred in the early 1990s, targeting severe combined immunodeficiency (SCID), commonly known as bubble boy disease. In this case, researchers used retroviral vectors to deliver a functioning copy of the defective gene responsible for the condition into the patient's own cells. This pioneering effort demonstrated the potential of gene therapy to treat genetic diseases at their source. With the advent of precise RNA gene editing technologies like CRISPR, gene therapy has evolved significantly, allowing for more accurate, efficient, and less error-prone methods of correcting genetic mutations, moving beyond the somewhat blunt approaches of earlier gene therapy techniques. 3. CRISPR-Cas9 in agriculture. The application of CRISPR-Cas9 technology in agriculture is another significant historical case demonstrating the impact of precise gene editing. Since its introduction in 2012, CRISPR has been utilized to enhance crop traits such as disease resistance, drought tolerance, and nutritional quality. For example, researchers have developed CRISPR-edited crops that can withstand harsh environmental conditions or have improved yields compared to their unedited counterparts. This rapid advancement in agricultural biotechnology parallels the medical applications of CRISPR, showcasing its versatility and the potential to revolutionize food production, much like it has the field of genetic medicine. These historical cases illustrate the transformative power of genomic technologies, from the foundational mapping of the genome to the precise editing of genes for therapeutic and agricultural benefits. They highlight a trajectory of innovation that has paved the way for the current and future applications of RNA gene editing technologies. Future outlook, predictions for precise RNA gene editing technology. The future of precise RNA gene editing technology, particularly through advancements in CRISPR systems, holds immense promise across multiple domains of medicine, agriculture, and biotechnology. Based on current trends and emerging technologies, several predictions can be made regarding the trajectory of this field over the next decade. 1. Enhanced precision and specificity. As research continues, we anticipate the development of next-generation CRISPR systems with enhanced precision and reduced off-target effects. Innovations such as engineered Cas proteins with improved PAM protospacer adjacent motif compatibility and high-fidelity variants will allow for more precise editing capabilities. This advancement will minimize unintended mutations, making gene editing safer for therapeutic applications. 2. Widespread therapeutic applications. The application of precise gene editing technologies in the treatment of genetic disorders will expand significantly. By 2030, we expect to see a plethora of clinical trials targeting various genetic conditions, including but not limited to sickle cell disease, muscular dystrophies, and inherited cardiovascular diseases. The success of these trials will likely lead to the establishment of approved gene therapies based on CRISPR technology, making personalized medicine a reality for many patients. 3. Integration with other biotechnologies. The convergence of RNA gene editing with other biotechnological advancements such as synthetic biology and nanotechnology will open new avenues for innovation. For example, the combination of CRISPR with gene circuits could enable dynamic control of gene expression in response to environmental stimuli, facilitating the development of smart therapeutics that adjust their activity in real time. 4. Expansion into agriculture. The agricultural sector will increasingly adopt precise RNA gene editing technologies to enhance crop resilience and yield. By 2030, we will likely see the widespread commercialization of genetically modified crops that are more resistant to diseases, pests, and climate change, thus contributing to food security in the face of a growing global population. 5. Regulatory developments. As the technology matures, regulatory frameworks will evolve to accommodate the safe application of gene editing technologies. By 2030, we expect to see clearer guidelines and standards set by global health authorities, leading to a more robust approval process for gene therapies. This will foster public trust and acceptance of gene editing technologies, essential for their integration into mainstream healthcare. 6. Ethical considerations in public discourse. The ethical implications of gene editing will remain a pivotal discussion point. As the technology becomes more accessible, there will be heightened scrutiny around its use, especially in germline editing. By 2030, we anticipate a more defined set of ethical guidelines and public policies governing the use of gene editing technologies, balancing innovation with societal concerns. 7. Advancements in delivery mechanisms. Innovative delivery systems such as lipid nanoparticles, virus-like particles, VLPs, and advanced AAV adeno-associated virus vectors will be crucial in enhancing the efficiency of gene editing technologies. By 2030, we predict that these delivery mechanisms will be significantly refined, allowing for targeted and efficient delivery of CRISPR components, particularly in vivo. This will be vital for achieving therapeutic success in complex tissues such as the brain and heart. 8. Evolving tools and techniques. The emergence of complementary gene editing tools such as base editors and prime editors will continue to evolve. These tools will allow for a broader range of genetic modifications without the need for double-strand breaks, making editing safer and more efficient. By 2030, we expect these technologies to be standard components of the genetic engineer's toolkit, allowing for detailed and nuanced genetic modifications. 9. Educational and research expansion. The rapid advancements in gene editing technologies will necessitate an expansion in educational resources and research training programs. By 2030, There will likely be an increase in specialized programs aimed at equipping the next generation of scientists and clinicians with the skills required to leverage these technologies effectively. 10. Global collaboration and accessibility. Global collaboration in gene editing research will become more prominent with international partnerships driving innovation and addressing global health challenges. By 2030, we can expect improved accessibility to gene editing technologies worldwide, particularly in developing countries, enabling broader applications and benefits. Conclusion. The future of precise RNA gene editing technology is poised for transformative advancements that will impact multiple sectors from healthcare to agriculture. With improvements in precision specificity delivery and regulatory frameworks, CRISPR and related technologies will likely become integral components of modern medicine and biotechnology. However, navigating the ethical considerations and ensuring equitable access will be crucial to harnessing the full potential of these technologies for the benefit of society as a whole.